This is lecture 8. In the last lecture, we talked about reflex arcs. In this one, we'll go over muscle fiber types, what determines their type, and switching between types. So what we know so far is how the brain recruits skeletal muscle, and we've gone over the macro and micro anatomy of the tissue and the cells it comprises. But not all fibers are the same. And in this lecture, we'll add some more properties to skeletal muscle. And then next lecture, we'll talk a bit more about the nerves that innervate them. I Googled muscle fiber types to see what came up, and it was this: type 1, type 2A, and type 2B. This information is brought to us by a website that has the word fun in the link, so maybe it's not meant to be serious, but in either case, there's a mistake here, and this one we'll discuss soon. But first, let's talk about type 1 and type 2 muscle fibers. Very generally, what you're flexing falls into two major categories, type 1 and type 2 muscle fibers. The type 1 fibers are darker in color, they're more oxidative in their metabolism, and they're slower and weaker in their contraction compared to the type 2s, which are lighter in color, less oxidative, and they produce stronger contractions. So the type 1s are better at endurance activities, and the type 2s are better in settings that require strength and power. So the fiber types, they sometimes get likened to the tortoise and the hare by way of analogy, but only to the extent of the Aesop's fable. In real life, turtles and tortoises are not endurance athletes. Right? There was a study published in the Journal of Morphology in 1995 that tested 7 hind limb muscles in turtles and found all of them to be at least half type 2. So a better analogy than the tortoise and the hare is probably a radio, where type 1 muscle fibers are AM waves and the type 2 fibers are FM waves. So the type 1 AM waves have a lower frequency. They can carry, that if you start driving out of distance, they'll follow you, right? They'll travel longer distances, but they don't pack as much of a punch. And then those type 2 FM waves have higher frequencies, right? They have more oomph, but they don't have as long of a range. You drive for 20 minutes and it gets staticky. So that's sort of how the type 1s and type 2s behave. But what does it mean in a practical sense? You can use your type 1 fibers all day long. They're really reliable hybrid cars. Let's do one more analogy. They have great gas mileage. They won't break down. They'll keep chugging along mile after mile, slowly, surely, conservatively, energy efficiently, but they won't do any speeding and they won't carry any heavy cargo. Those are your type 1s. If you want to speed or do hauling, you need a muscle car, you need type 2 fibers, huge engines. The power to weight ratio, the horsepower to girder structure ratio is much higher in type 2s, but they're not meant to be driven in school zones. Okay, so 3 analogies, that should suffice. Let's move on. Let's move in reverse. Let's go backwards to this, which is the Google search I did for muscle fiber type. We have type 1, type 2A, and type 2B. What's the problem here? The 2Bs. That's the problem. If you see a bunch of 2B fibers in your tissue, then you have the problem. Unless you're a quadruped who learned to read, or Splinter from the Teenage Mutant Ninja Turtles. 2B fibers are all over the place in rats and in plenty of other non-human animal species, but it's not really in us. I think there's tiny bits of it reported here and there. It's been observed in laryngeal muscles, but it's not really expressed in us. You're not going to find the protein, this particular protein in your limbs. It is fast, though. It's a very, very fast twitch muscle, which means a type of myosin heavy chain permits rapid cross-bridge cycling. But we have other fibers to take care of our nearly that fast contraction needs. According to the Essentials of Strength and Conditioning, these are your fiber types, 1, 1C, 2C, 2AC, 2A, 2AX, and 2X. Not just plain old dark meat and white meat, but 7 shades of meat in all. And We've talked about types 1 and 2 very generally, but C, A, and X specifically, those matter too. C, less so. If you sample the muscles in your limbs, you'll find a small amount of it. It's an immature myosin form, and you'd expect to see it in regenerating fibers. The main fiber types to pay attention to, though, here are 1, 2A, and 2X. And each of these has its own myosin isoform, its own version of myosin heavy chain, which changes the performance of the muscle fiber. Looking at myosin here, a single myosin molecule has two heavy chains and four light chains. So often there's only one myosin heavy chain type in a muscle fiber. It's type 1 or it's 2A or it's 2X. Purebred muscle fibers, a pure distance running fiber, a pure sprinter fiber. Um, you see the light chains, those are the necklaces around the heavy chains' necks. Essential light chain seems to be more about structural stability of myosin, and the regulatory light chain is a bit more involved in the contraction itself. Now, we do see more types of sarcomeric myosin than we've accounted for, right, by our 1C, 2C, 2AC, 2A, 2AX, 2X model. The Human Genome Project identified these 11 different genes that express myosin heavy chain. And here's another look at the same thing. Among this variety of myosin heavy chain, some of it is embryonic. And being adults who are listening to this, what we really care about is adult forms of the protein. And 2B is on here also, and we don't really have that in a notable amount. At the bottom of the table, you see there are three forms of non-muscle myosin beyond the 11. And this isn't for human movement, it's for cell adhesion. And you'll notice in the highlighted text at the bottom of the paragraph, it says, within myosin 2s found in human muscle, there are 11 different sarcomeric myosin isoforms, two smooth muscle isoforms, as well as three non-muscle isoforms. What it means by myosin 2 is not 2A and 2X. Myosin 2 just means the type of myosin you're thinking of. It's sometimes referred to as the conventional form, but it's the kind that's responsible for contraction in a muscle cell, as opposed to type 1, which has functions in vesicle transport and some inner ear rolls. But we have a lot of myosin heavy chain isoforms. Here's a similar breakdown, but you see different light chains represented here as well. There are different isoforms of myosin light chains too, which means different versions of those proteins. It's not just heavy chain differences we see. In this table, we're looking at heavy chain and light chain differences again, but you see 2B there, they're looking at rats. But in humans and rats and all other critters that move, both myosin heavy chain and light chain influence contraction velocity. That's not conduction velocity of the nerves. We'll talk about that in the next lecture, but contraction velocity, meaning how quickly cross-bridge cycling cycles. But each of these proteins comes from a different gene. This isn't like one version of myosin heavy chain being translated, being synthesized, and then all of the variety being produced by post-translational modification. That's not how this works. There are distinct genes making the distinct proteins. But with the mosaic fibers, what this means is that not all muscle fibers are purebreds. They're muscle fibers that are purely type 1, right? Meaning that their form of myosin is type 1. There are muscle fibers that are purely type 2A, meaning that's their form of myosin, and the same with 2X. Like if you buy wine, you can buy a bottle of Pinot Noir or Zinfandel or Malbec. There's a specific type of grape in each, right? Zinfandel is made from Zinfandel grapes. So you can buy a pure grape bottle, or you can buy a blend, right? Malbec and Cab Sauvignon and Merlot, all mixed, right? You can buy those blended bottles too. And the same thing is true with muscle fibers. By mosaic fibers, what people mean is that individual muscle fibers aren't all purebred, right? Some of them are hybrids, some of them are purebred, but some of them are also hybrids, which explains why 2A is a muscle fiber type and 2X is a muscle fiber type, and 2AX is also a muscle fiber type. The velocity of a muscle contraction is determined by both myosin heavy chain and myosin light chain. But the metabolic part, the energy consumption, that's exclusively determined by the heavy chain. And the reason I have a magnetic bead necklace doing the speaking here is that it's a reasonable illustration of how proteins work, proteins in general, and myosin is one such protein. Each bead in the necklace is an amino acid, and the translation of a protein is the linking together of a string of amino acids. And depending on how the protein is made, which amino acids are linked in the series, the protein folds differently. And how it folds determines its behavior. So if you ever put a magnetic necklace on a table and watched it crinkle up, proteins crinkle too. And if you barely change the beads, that necklace will fold differently and it will, behave differently. So that's how we have different genes creating different versions of these proteins and how that affects their behavior. Now, the physical differences in the myosin protein, how it folds and creates differences, the differences we're talking about is the lever, right? The lever itself changes, the length and the shape of the lever changes. And if you change that, you change its performance. There are functional differences between myosin heavy chain types. The slow fibers, those they can maintain isometric tension effectively and very efficiently. But the type 2A and 2X myosin forms are necessary for speed and power. The rate of contraction varies enormously between these, by a factor of up to 10. But the stronger, faster myosin forms, those also consume much more ATP. Right, getting back to this idea of how we categorize the muscle fiber types, this is another textbook stratification. You see the ones and twos, CAX, and some developmental heavy chain forms. Now, you don't need to know anything here specifically, but before we move into the practical matters, it's reasonable to wonder how we classify these. Historically, there's been a variety of approaches. Slow and fast myosin isoforms could be chemically distinguished because of the different amounts of ATP they consume, the different rates of ATPase activity. The histochemical methods can be sort of stumped though with hybrid fibers. The fibers have different versions of myosin heavy chain in the same fiber, but there's other stuff to look at too. You can compare aerobic and anaerobic traits of the type 1s and the type 2As and 2Xs. So more oxidative fibers are going to have more mitochondria and lipids and capillaries, right? And myoglobin. And there are other structural differences too. Like in type 1 fibers, the sarcoplasmic reticula are less abundant, and the version of ATPase that pumps the calcium back into the reticula following a contraction, that's slower. And then in more anaerobic fibers, there's more glycolytic enzymes and glycogen, and the sarcoplasmic reticula are denser in those. Okay, let's get more general. The three skeletal muscle fiber types we really talk about are these: type 1, type 2A, and type 2X. But it's not just the myosin that determines the speed of contraction. Qualities of the sarcoplasmic reticulum are also important. That's not something we'll be getting into in detail here now. But other than myosin heavy chain isoforms, this stuff matters. The stuff on the slide here matters. We'll get into the motor neurons more in the next lecture, a little more today, but ATP matters too. Different myosin heavy chains consume ATP at different rates, and the consumption of ATP results in ADP and phosphate levels rising, which can impair the rate of cross-bridge cycling. We'll talk about that more at the end of the semester. But this is just to say that myosin heavy chain gets most of the attention, and deservedly, but it's not the only determinant of contraction velocity. Now, all of the fibers that live within a single motor unit tend to be similar in type. Some motor units are populated with type 1 fibers, Others are populated with type 2A fibers, and other motor units have a bunch of type 2X in them. But each motor unit can be thought of as a household filled with family members, right? Siblings and aunts and uncles. And in a household, most people, they're going to speak the same language, okay? They're going to hold the same customs. They're going to be similar. And the muscle fibers behave this way too. An individual motor neuron will activate slow twitch fibers or fast twitch fibers or even faster twitch fibers. Motor units aren't very diverse in their fiber populations. This is a look at some of the differences in properties and behaviors between types 1, 2A, and 2X. There's overlap here between entries, yeah, like you see endurance and fatigue resistance. Those are kind of describing the same thing. But when you keep looking, you see other variables describing endurance too, like aerobic enzymes and capillary density and mitochondrial content. But overall, this is a nice look at some general differences in these fiber types. All right, so now that we know about the different muscle fiber types, can we switch them? Or are we just born with an assignment that we have to accept? The answer to both those questions is yes. In the same way that we can ask, can older adults switch political parties, or are they just stuck in their mindsets? The answer to both of those is also yes. People don't readily switch, but they can. So on the left here, we have Democrats. On the right, we have Republicans. We have a two-party system. But within those two parties, there's plenty of diversity, right? There's different sub-parties. Like libertarians are right aligned, and the Green Party, that's left aligned. So there will be purebred Republicans and purebred Democrats. But there are also hybrids across the spectrum, and the same is true with muscle fibers. And with a lot of use of using those fibers, you do see a characteristic migration from 2X to 2A. To an extent, all muscle fiber use is an oxidative event. Unless there's a skeletal muscle infarction during your set, there's oxidative activity happening. And you do see some changes in characteristics to support that, some chronic changes to support it. But this is like migrating within the political party. You favor a more or less progressive Democrat in the next election, or a more or less conservative Republican. You know, a constitutionalist votes for a traditional Republican, a Green Party member votes for a traditional Democrat. But switching from elephant to donkey or vice versa, this is a much bigger change. And until medicine advances to a state in which brain transplants are possible, it's probably going to be a rare change. And the same is true with skeletal muscle fibers. Except the difference is brain transplants happen all the time, right? It's called axonal sprouting or neuronal sprouting. Exercise-induced damage, like if you're running all the time, that'll occasionally result in neuronal detachment, and a new neuron can pick up that fiber from the lost and found. And remember, a motor unit tends to be populated by the same fiber type, or at least the same fiber behavior. So if it's just picked up a stray, that stray will take on characteristics of the new household. It learns the language and the customs of the head of the household. In this study, the authors used 20 rabbits and they surgically swapped fast and slow nerve attachments. This is called cross reinnervation. They surgically attached different motor nerves to the fiber, and they found physiological and biochemical differences afterward. In a way, this is an expression of specificity of adaptation. Form follows function, and nerves control that function in nature, through exercise and normal daily behavior, cross reinnervation will happen on occasion. So think of it like a nerve picking up fibers at the lost and found, right? If there's a pair of shoes that no one is claiming, you can go claim them, finders keepers. Now, maybe they're basketball shoes and you're not a basketball player. So the basketball player who lost them gave up their assignment. And the person who uses them now, the person who claimed them, maybe they're a runner. Are the shoes perfect for running? Are basketball shoes good for running? Well, no, but you'll break them in, right? Those shoes will start to take on characteristics of running shoes. And that's how muscle fibers work. That's true with muscle fibers. Now, does everything change inside of the fibers? Um, is a fiber that underwent neuronal reassignment indistinguishable from a fiber that was born that way? Not quite. Close, not quite. Think of it like gender reassignment for all emotional and social and psychological and practical and all ethical purposes. Caitlyn Jenner is 100% changed. But if you were to inspect the chromosomes, relics of Bruce can be detected. Um, and that presents a good analogy for fiber type changes of this kind. For all functional purposes, yes, they've completely adopted their new environment, but you're likely to see remnants of its former phenotype. Now, remember that in a single motor unit, most of the fibers aren't neighbors, right? They aren't immediately adjacent to one another. When they're like widely distributed in this way, the contractile force of each motor unit is more evenly spread throughout the whole muscle. But after axonal sprouting, neuronal or axonal sprouting, neighbors often start to form. So you might see some of that random scatter become more clumped together. But not all motor units are the same. Their range is different. Their general pattern of geography is different. Each on average, each one, maybe it covers 10%, give or take a ton of the muscle's total cross-sectional area. It's not covering 100% of it. But when you see clumping of this kind, the kind pictured here, type 1 fiber grouping, it may be caused by conditions like Parkinson's, which can result in the deinnervation of type 2 fibers and then the subsequent reinnervation by the type 1s. In this case, if you read the last sentence there, the motor unit takes on at least some of the type 1 phenotype, including expression of the new myosin isoform. And environmental stresses can impact this too, like microgravity, go up in outer space. That can decrease type 1 fibers. In rats, a couple of weeks in microgravity resulted in a decrease in the percentage of type 1s, while the percentage in type 2s increased relative to that. But keep in mind that in humans, there's some give or take in our estimations. Biopsies don't evaluate the entire muscle, so changes are just that, they're estimations. They're not perfectly accurate assessments. So what percentage of fibers do we have? In the diagram to the right, you can see a type 1 fiber, um, the dark one, right? A type 2X, the paper white one. And there's a type 2A, that's the tan fiber. And in the table to the left, that breaks down fiber type percentages in some muscles throughout the body. It's just type 1s compared to type 2s. The orbicularis oculi, for example, that's only about 15% type 1. So a really high percentage of type 2s. It's a blinking muscle. You blink explosively, right? And then you rest. And then you blink explosively again. You blink fast, right? And then you rest. You don't blink for 200 reps in a row. So there's no point in having a bunch of type 1 fibers in there. The triceps brachii, triceps up in your arm, maybe half and half in that one. The soleus, that's the lower right muscle in the table. The sandal of your calf, that one is about 80% type 1. So the muscle fiber, the composition varies between muscles. It also varies based on the individual in a single muscle. So this study here, that's the vastus lateralis, a quad muscle. And there are 19 different young males who were tested. One of them only had 32% type 1s, meaning 68% type 2 fibers. And another subject had 76% type 1s, meaning 24% type 2s. So people vary. And if you want to be an elite sprinter, you need a high percentage of type 2s. If you want to be an elite endurance athlete, you need a high percentage of type 1s. And where you're plunging the biopsy needle into the tissue, that matters too. Deeper into the tissue, you're likely to pick up a higher relative percentage of type 1 fibers, whereas the more superficial portion of the muscle, that's likely to have more type 2 fibers comparatively. This is a glimpse at individual muscles and some estimations of composition in the deep and superficial portions. So imagine you were to take a biopsy and just scrape it all over your limbs, like it's a liposuction vacuum, right? And you're just sucking up all of your muscle into your myosuction vacuum biopsy needle. And you ran that needle's worth of contents to test your muscle fiber types. It would be somewhere near half and half, right? Half type 1, half type 2, erring on the side of type 1. But deeper into the muscle, it's more concentrated with type 1 fibers. And maybe it's so the shell is more protective, right? You can fast, hard flexion to brace against impacts or something. Maybe it's to minimize friction, right? The more superficial the fiber, the more it rainbows through its line of pull. Maybe it's about heat dissipation, more heat produced in the periphery is easier to offload. Maybe it's one of 50 other explanations, the reason we see this pattern, but it is well represented. The deeper you go, the more of a type 1 creature you become. So, since we have different muscle fiber compositions, you, me, and everyone else, what athletic contexts associate with each? If your muscle looks like this, all dark meat, you're going to be metabolically efficient, meaning more work done with a given number of ATP consumed, but you'll be weak and slow. So maybe not big power exercises, but you can go all day, right? So activities that don't require sprinting or high jumping or heavy heaving would be best, right? Long swims, long jogs, long bike rides, long anythings. All the way over here, it's all white meat, right? Lots of type 2 fibers. When you need to be fast and powerful, right? You're running from a predator, you're chasing prey, or you're competing in 21st century equivalents of those, right? Anaerobic sports, you're doing the 100-meter dash, your 200 meters, or you're weightlifting, football, right? Like American football. You're pitching, or any other throwing sport. These are likely to come naturally to you if you have a natural abundance of type 2 muscle fibers. But in the middle, we have hybrid sports like soccer, hockey, lacrosse, basketball, sports like this, or like a mile run where you need to kick at the end. If that kick matters, having some type 2s is going to help you achieve a mighty kick. But now, how do we decide what kind of muscle fibers to recruit? The type 1s or the type 2s? Like, let's say you have roommates and it's in the middle of the night and you decide you're thirsty and you like milk is what you're thirsty for. What determines how much force you put into your milk hoist? I mean, do you fail to lift it? Do you smash it into the top of the fridge? The number of motor units you're calling upon is really what's determining what muscle fiber type. And by extension, the muscle fibers being recruited, right? Not just how many, but the type. So notice that gastrocnemius in this chart. Scarcely are you doing fine motor coordination with your gastroc, right? You're moving a big body around, and you need strength for that, not motor control. So you have a couple thousand fibers in a motor unit. In the biceps brachii, maybe you have 700 or 800 fibers per motor unit. So this ratio depends where you are in the body, how many fibers per motor nerve. And you know rate coding, we've covered this previously, but we'll cover it again briefly. On the y-axis, that's your tension, how much tension is in the muscle, how much is it generating. And on the x-axis, that's time. Is it one little action potential, one little stimulation, or is it one and then another and then another? Or is it a ton of stimulations in a row? So in addition to the number of motor units you recruit, um, how many muscle fibers are in each motor unit, and the muscle fiber types in those units, rate coding contributes to force generation. This is just another look at it, where you can see the myoplasmic calcium corresponding to the firing frequency, the frequency of action potentials. And in the next lecture, we're going to return to the neuronal component of muscle control, and we'll talk about the Henneman size principle. But for today, here's what you need to know. These questions will summarize the lesson if you can answer them. And I will see you in lecture 9 when we discuss orderly recruitment or Henneman size principle.